I'd like to introduce our next speaker. He's Dr. Jed Johnson. He's with Nanofiber Solutions, and I'll let him tell you about the company. Jed knows a little something about nanotechnology. So,
great. Thank you.
Jed.
Thank you. Great for the invite, and I hope to kind of equip some of you today. But I always like to do a little audience participation. So who here is a MD? Raise your hand. Okay. And then how about engineer? And then we'll do administration, business, everything else. Okay. All right. Just curious. So I'm an engineer by training, graduated from Ohio State. And so I'll talk a little bit about nanotechnology. And what I really want to do in my talk is to encourage you to think outside of the box. Engineers in particular like to have a pretty narrow focus, and we can really be trapped into not seeing other applications for some of these technologies. So some examples of situations like this: sticky notes from 3M. That was actually a failed product. They were trying to come up with a super strong aerospace adhesive, and I'm pretty sure today we would call that a great success. GPS is another example of a design for DoD, and now we use it to find Taco Bells, or as we're shopping through IKEA, to tell us about the display units. And then nylon, which was created for a silk parachute substitute, and now probably half of this room is made out of nylon. So really think outside of the box of some of the examples that we're presenting today. I'll talk a lot about nanofibers. That's just something I know a lot about, but really think other applications and other uses of some of these technologies. So since we're not all engineers and we might not always know what some of the prefixes are, a nanometer is a pretty common question. And we always use the term nanotechnology, but what does it really mean? And the example that I like to give is this Coke bottle example. So a Coke bottle is made out of polyethylene terephthalate. It's also the same thing that's made in Dacron vascular grafts. It's just a smaller form of that. Those fibers are about 50 microns in diameter, close to a human hair in size. The fibers that we make here on this right side are in the nanometer range. So 500 nanometers, that's about 100 times smaller than the width of a human hair, so very small. It's exactly the same material, though, from a Coke bottle to a Dacron vascular graft to some of our nanofibers. And the whole reason we do that is if you think of a typical cell size, a red blood cell is about 8 microns in diameter. They need something that size or smaller to really grab a hold of and interact with. So our 500 nanometer fibers are equivalent to us grabbing a baseball. We can grab a baseball and we can throw it. Something 100 times larger than a baseball is a house. So a cell on these traditional Dacron vascular grafts, the cell really has trouble grabbing hold of those fibers, and it essentially sees a flat surface. So if you could imagine trying to pitch a house, that's the difference between, say, microtechnology and nanotechnology. So you might not know it, but in nanotechnology, in current medical devices, or in just general use today, you probably use it every day, and you have no idea that you're using it. So everything from diamond-coated blades, these are scalpel blades, needles, even pocket knives, to bone fillers, hydroxyapatite and beta-tricalcium phosphate. Shark skin and other antibacterial coatings have a nano-textured surface to prevent biofilm deposition. Cosmetics and sunscreen, so as you apply sunscreen to your kids, it's full of nanoparticles. So some pretty common everyday things. It's not always really high-tech, really expensive medical devices that use nanotechnology. And a lot of point-of-care diagnostics are being utilized today with nanotechnology that weren't available before. So with the smaller size, the larger surface area, we can really reduce the size of the samples that we collect. So now from a saliva test, we can start to diagnose cancer in patients. So tremendous things that we weren't able to do before, where we had to take large samples and large times to measure those samples. Now we can do in minutes with drops of saliva, blood, or urine. And then a lot of drug delivery, different modalities. So whether it's enabling ultrasound imaging with microbubbles or nanobubbles, to thermotherapy, where we can deliver particles to tumors to accelerate thermal heating of those. And then the example that we always like to show, just because it's nice and colorful, are these solutions up here. Believe it or not, those are all gold particles. And we always think of gold being the gold color, but due to the nanotechnology and the way it interacts with light, we can actually change how we perceive that. So a lot of materials behave much differently on the nano scale than they do on the bulk or the macroscopic scale. So what we do with nanofibers, we use this process called electrospinning. We take the polymer, any FDA-approved polymer or any kind of homemade polymer, we dissolve it in a solvent, and then we apply a very large voltage to this solution, and it ejects the stream spiraling down here. So the stream, then we can collect onto a substrate, and it forms this fibrous mesh, very much like a paper towel or a napkin. It's almost like a textile, except these fibers are those 500 nanometer fibers that I showed earlier. So the whole reason we do it is that if we take a look at decellularized tissue here on the left, this happens to be a blood vessel. I have pictures of skin, of fat, of cartilage. And when you strip off the living cells, what's left is this fibrous protein structure. The fibers that we make are here on the right, and you can see immediately that imitation there. So we have the same topography, the same structure. And so the only reason we're making nanofibers is to mimic what's found in the body. There's different parts of the body, like white matter in the brain and myocardium from the heart, skeletal muscle, nerves, that all have alignment and orientation. So if we try to look at reproducing those structures, we need to reproduce that alignment and that orientation. So we can create these aligned fiber structures here on the right using the same synthetic materials. And then as a company or as a technology, we can really do a lot with the different fibers. So we can make truly small nanofibers, so these are less than 100 nanometers in diameter. We can go up to more microfibers, so those are about 20 to 30 microns in diameter, which is very similar to the clothing and synthetic clothing. We can do core shell fibers there on the right. So if you think of a coaxial cable that has a rubber outer casing and the metal going through there, we can do very fancy things like that on the nano scale. We can add in nanoporosity onto these nanofibers. We can also embed all sorts of particles, whether these are hydroxyapatite for bone, or these can be drug particles, really all types of things that we can put into the fibers. And so what can we do with this? So this is kind of going outside of that box concept. So if you think of fibers, that's great. You probably think of textiles, of clothing, but some of the applications that we work in are cell culture dishes here on the left, which can be from pharmaceutical drug discovery applications to stem cell assays in a lab, all using this normal multi-well plate. To here in the middle, this is a GE Wave bioreactor, and these are used for industrial stem cell expansion or production. So a lot of companies today are growing stem cells as their therapeutic. They're producing millions of cells per patient and ideally treating millions of people across the world, so that we need trillions of cells to treat that many people. And how we do it is with high density cell culture. So we have these bioreactors and we can grow trillions of cells in a small plastic bag instead of using huge warehouses to produce these cells. And then here on the right is an example of a trachea and tissue engineering applications where we're actually implanting these nanofiber scaffolds into the body to replace or repair different structures or organs inside of the body. So how did we come up with some of those different ideas and those different applications? Well, interdisciplinary collaboration. So in this picture shows engineering, shows medicine, shows neuroscience, and the pharmacy. So a huge team brought together to develop some of these applications, whether it's drug discovery on the pharmaceutical side or brain tumor treatments on the neurosurgery side. So it takes a lot of different experts from different fields to come together to develop some of these products. The days of engineers developing things on their own are gone, and we really need to bring in areas of need and expertise across the board. So I'll segue a little bit into just some examples of the fibers and then different applications that we do with those based on this collaboration concept. So Nino Kioka was a neurosurgeon here who helped us develop some of our first products And here we were looking at a way to see how glioblastoma or brain tumor cells migrate across the fibers. So if we place one of these tumor cells on the fibers, they kind of round up and look something like this. If we place the same tumor cells on the line fibers, these cells really elongate and form this invasive phenotype here, and they become very invasive, very migratory, and published a paper showing that they're about 5 times more migratory on these fibers. So with that, one of the examples of what we did with that is this video here. So we can take a tumor biopsy, label it with a dye, and then put it onto these aligned fibers. And then you can see the time advancing in this upper left-hand corner. And over the course of about 24 to 36 hours, these cells are moving millimeters. They're incredibly invasive, and this is one of the things that make brain tumors so deadly and so hard to treat. So with this simple experiment, they were able to run tests that they were never able to do before. So as part of my PhD work here at Ohio State, this is some of the work that came out of it. So, and this would only be possible by this interdisciplinary collaboration. Another example, working with Clay Marsh, is with idiopathic pulmonary fibrosis. So here we're looking at recreating this disease in a dish. So we can take normal lungs and we can take fibrotic lungs, apply them to the fibers. Then we can seed stem cells on top of that mixture and then look at how those stem cells are differentiating in response to the diseased state or the normal state. Another example of the tissue engineering now is working with Gail Besner at Children's Hospital. So here her focus is necrotizing enterocolitis in babies that are developing short bowel syndrome. And currently there's no technique to transplant a intestine for these kids. So the ability to be able to grow an intestine for these kids changes that. So what we do, create a tubular scaffold, and then we can seed it with stem cells and then implant it with an end-to-end anastomosis in line with that intestine, and we can regenerate that intestinal structure. Working with Chris Brewer, also at Children's Hospital, on arterial grafts. So here, the current technologies of Dacron and Gore-Tex are limited, that they don't grow. So specifically with the pediatric population, those grafts stay the same size and the patient continues to grow. So they have to be removed and replaced with other technologies. So with this nanofiber technology, we can implant grafts that can fully resorb or degrade over time and be regenerated with native tissue. So this would allow the blood vessels to grow as the patient grows, as well as to address some smaller diameter vascular grafts that current technologies can't do. And this is a short trailer I'll show. This is a trailer from a documentary that was made on the tissue engineered trachea. This was developed in collaboration with the Karolinska Institute in Sweden, and just further demonstrates this collaboration. So I'll show this. It's about a minute long.
Probably everybody of you have
Hopefully
seen
you can hear
the
that.
patient. She's a wonderful girl that unfortunately has had a car accident during the 6 months of pregnancy, for which, among other things, she has a complete transected airway. So tomorrow, we plan to do the first in human ever done laryngotracheal transplants using bioartificial scaffolds.
So we take these CAT scans, CT scans of the patient's neck. From the dimensions, we make this replica in plastic, the exact replica of the patient's windpipe or trachea. And the stem cells are seeded, almost rained down on top of the scaffold inside the bioreactor.
Und wir glauben, dass es in naher Zukunft möglich sein wird, Patienten maßgeschneiderte Organe zur Verfügung zu stellen.
What we're doing today, people never thought was possible. What we've been able to accomplish has changed how medicine has been thought of.
Tomorrow, 2 of the best airway surgeons in the world will operate together. If we don't achieve this, who should do it?
So just another example of this collaboration of where that surgeon was using decellularized tracheas from cadavers to implant into patients, and now we can come up with a fully synthetic implant for that. So without that collaboration, we would never be able to come up with a trachea on our own, and they would never be able to come up with a synthetic option for a transplant. So just to further demonstrate that, so all those different examples all feature the same polymer fibers, but now we're showing a whole range of different applications and uses. So as you have ideas or technologies or concepts, you kind of need to have this game plan or a strategic plan laid out of what you want to do with it. So this is a very busy slide. Don't try to read all of it, but the basic idea is that you have today and some applications that you can go after with your technology. So for us, this is three-dimensional cell culture, some drug discovery applications, and then within, say, 2 to 5 years, we have some acellular products. So these are simple hernia meshes, wound care, that type of thing. If we look out, say, 7 to 12 years, we start to look at tubular organs. So these are the blood vessels, tracheas, that sort of thing. And then you can keep going on. So to have a strategic plan in mind for your technology and have a roadmap of what you're addressing. And similar to the previous talk with the process for design and development, you need a process for commercialization and how you're going to target that. So I wanted to close with some resources to help you. You're never going to have everything that you need in a 20-minute talk, let alone a half an hour a day or a half a day. So I wanted to leave a couple things with you that have personally been very helpful to me. And some of these are long URLs, but Scott or Don, or you can contact me. One of us will be able to get some of these to you. But National Council of Entrepreneurial Tech Transfer, they have a series of weekly webinars that cover everything from intellectual property to idea creation and commercialization, business plans, marketing, across the board. And this is a federal agency. It's all free, and it's all very well done. Tech Columbus, which is right up the street, which is where we are currently located, has a tremendous amount of resources to help you commercialize technologies. So as you have ideas, if you have prototypes, as you have negotiations on the table at all levels, they can help provide manpower and expertise to help you along those lines. They can also provide incubator space. So if you need incubator as in business incubator, not cell culture incubators, have to be a little clarifying there. And then they can also help provide funding. So small grants that start at $50,000 on up to more sophisticated investment vehicles such as convertible debt, convertible loans, approaching $500,000 to $1 million. So certainly a good source. Many of you have probably seen this, the FDA Center for Devices and Radiological Health. The FDA's website has a tremendous amount of information. It's also probably one of the hardest to navigate that I've ever seen. But CDRH Learn, if you just Google CDHR Learn, has a series of modules that really help show you the different regulatory pathways. So if you have a simple device that can go through a 510K pathway versus an artificial blood vessel that is going to be a PMA product with years of development, they can help you learn those different paths. U.S. Patent and Trademark Office, also a terrible website to navigate, but with tremendous amount of information. If you Google the IP Learn or eLearn, they have a couple of webinars that walk you through basics of patenting, so you can know the difference between provisionals, non-provisionals, design patents, and on and on. And then my last two here, I'll bring both of these up here together. Project on Emerging Technologies, and then this is actually a Netherlands project, RIVM, there at the bottom. Both have a lot of topics and information on the merger of nanotechnology and medical devices specifically. So if you want to see what other technologies are out there or some additional resources on nanotech and medical space, those are two excellent resources. So with that, thank you.